Hello, and welcome back. This is Professor Young, and we are dealing with part 3 of 3 of our look at cellular biochemistry. So far, we've done a basic review of chem. We've talked about carbohydrates. We've talked about nucleic acids. And in this session, 3 of 3 again, we're going to be really focusing in primarily on proteins and the role that proteins play throughout the body, which is a fairly substantial role. And then we'll be spending a couple minutes just kind of highlighting lipids. And so why don't we go ahead and get started. So far, every major macromolecule we have looked at it from the simple perspective, and we've also looked at it from the complex. This is no different with the proteins. When we were dealing with carbohydrates, we know that monosaccharides, which are the simple sugars, are linked together through dehydration synthesis to form disaccharides and polysaccharides. We know that from looking at nucleic acids that nucleotides are the base or the simple structure that will join together to form a nucleic acid, that being DNA or either RNA. Again, that's going to happen through dehydration synthesis. And we again see this with proteins. Proteins are the macromolecule, but they are also made up of subunits. And those subunits that we are referring to are called amino acids. We're going to talk a little bit more about amino acids once we jump into Chapter 4. But for right now, what you need to make sure that you understand is that there are 20 different amino acids that the human body relies on, and that these amino acids are basic building blocks for proteins. In other words, as we link amino acids together through dehydration synthesis, we go ahead and form complex proteins. Of the 20 amino acids that we have within our body, 11 of them are made within the body. In other words, as you take in food and energy, the body can transform them into the 11 of the 20 amino acids. There is another 9, though, that are referred to as the essential 9. And they're considered the essential 9 because our body does not have the ability to produce them on their own. They need to be acquired through dietary habit. Now, all amino acids share a very common structure. All amino acids start out with a central carbon atom, which you can see right here. Right? They all start out with a central carbon atom. They all have or maintain a carboxyl group right here on the ends. Right now, notice there is a difference between those two carboxyl groups. In the top one, we have a straight up hydroxide ion that is bound to the carbon, which is double bound to an oxygen. That is the standard. That is the typical. There is some change that will occur, especially once the amino acid has gone through dehydration synthesis and has bound to another amino acid. And that change is it will lose a hydrogen and just become a O negative. Both of these are carboxyl groups. They're just different versions, if you would, of the same thing. So don't get confused by that. We also have a hydrogen atom that'll be present on all amino acids. And we also have an amino group. Right now, again, notice there's differences between those amino groups. One's an NH2, one's an NH3. As long as there's a nitrogen bound to hydrogen, it's going to be an amino group. So don't get bogged down whether it's an NH3 plus or an NH2. No big difference there, at least not for our understanding. Right? It's still an amino acid. And lastly, we also have an R group right there and right there. And those R groups, that is what really distinguishes one amino acid from another. That is where all of the action is going to take place. That is where all of the behavioral characteristics are found within an amino acid. If we take a look at this chart here, again, you don't need to memorize this chart. You don't need to know the structures necessarily. But what I do want to point out to you that is that if you look at each of the main components of these amino acids, they all are that predictable characteristic. And they all share that basic formula that you saw on the previous slide. What does change for all of them, though, are the side chains. If you look, I'm trying to see what the best way to do this is, if you look right there, there's one side chain. There's a different side chain. Here's yet another different side chain. Here's yet another different side chain. Here's yet another different side chain. All of these have varying side chains. And that is really going to influence how that amino acid behaves. It's going to determine how those amino acids are going to react with water. It's going to determine whether or not it can react with water. Maybe that side chain is a lipid-based side chain, at which point in time it is not going to be water soluble. It is going to be, indeed, hydrophobic. Right? Maybe it is a water soluble. Side chain, and so therefore then it will be hydrophilic. The bottom line here is those side chains do have an impact on the ability for the amino acid to react, and it will alter the solubility potential of the amino acids. And so that's critical for you to understand right there. Now, we talked also a little bit about the fact that amino acids are that base unit. They are the simplest molecular unit for a protein, whereas monosaccharides is what we refer to as the base unit of carbohydrates. Here, we just simply use the term monomer. A monomer is an amino acid. It's the simplest molecular unit that will make up a protein. When we go through and do the process of dehydration synthesis to link amino acid to amino acid to amino acid to amino acid, we then refer to that process as polymerization. Polymerization is the process of linking amino acid to amino acid using dehydration synthesis. Polymerization is the process of linking amino acid to amino acid to amino acid using dehydration synthesis. All right. And again, another word or another term that we can substitute here for amino acid is monomer. They're synonymous. They're one and the same. Now, once we have linked 50 or so, 50, 100, 200, 450, 1,000 of these amino acids together, then what we have created is what we refer to as a macromolecule. We've created, in a sense, we've taken that macromolecule and we've created the protein. And so again, this here is a slide that's just going to review that. You can see up top here, right here in this section of the screen, you can see that as those monomers are coming in, as those amino acids are coming in, they are being linked together. That is the process of creating a polymer. That is polymerization. We are using dehydration synthesis to form those bonds. And so this here is dehydration synthesis at work. Okay, that is dehydration synthesis. If we wanted to split these guys apart, if we wanted to split them apart, then we would use hydrolysis. The bond that is holding these monomers together, are what we refer to as a peptide bond. Right? And as we will see on the next screen, we are forming that peptide bond between the carbon of one amino acid and the nitrogen of another amino acid. Think about it. What is bound to that carbon? It's a carboxyl group. And so right away we know that we have to at least have a hydroxide ion that is more than likely going to be bound to that carbon. And if we take this a step further, what is bound to the nitrogen? At least two hydrogens, maybe three. And so if we take the hydroxide group from the carbon and a hydrogen from the nitrogen, we basically essentially have done dehydration synthesis. And what we are left with is, we're left with that peptide bond. Now, the other thing that can happen here is, no, I won't go there. I'll leave that all in. So we'll take a look at that there on the next slide, which is coming up right here. Right, so we can see here we are removing water. So a smaller one right there. Here we are removing water, and as we remove that water, we form the peptide bond right there. And the next picture, right here, we are adding the water back in. We're adding water back in, and as we add that water back in, again, hydrolysis, we are splitting apart that peptide bond. So this here would be hydrolysis. This here would be dehydration synthesis. All right. And you can see what's happening down in this area here as far as how we go about doing dehydration synthesis. Again, we're using, in this example here, we're using an ulterior or an alternate form of the OH. All right. Instead of having a straight hydroxide group here or a hydroxyl group here, we just simply have the O negative. And so we're taking that. And because there's not an OH on the O negative, then we also need to have two hydrogens that are removed here. And when we remove all three of those guys put together, when we remove those three components right there, we have released the water. And then the bond is happening or occurring to that double bound oxygen right there. So that is how we basically link amino acids together to form these proteins. Now keep in mind, there are, I think it's been estimated, something like 15 to 1,700 different proteins that we have within the body. All right. Those various proteins all have varying functions. And the functions are related to, as you can probably guess, the structure. This is a unifying theme that kind of goes through all of anatomy and physiology. Structure dictates function. And that's what you're going to see here on the next slide. Oops, I lied. On the next slide, we're going to see how the structure impacts the function. On this slide that we are currently on is an overview of exactly what proteins do. And you can see here that proteins do a very wide array of tasks within the body. Probably the most important one being enzymes. And we're going to talk about enzymes coming up here in a couple of seconds because they are critical in the whole process of metabolism and how we break apart energy and how we store energy and how we utilize that energy. So enzymes are going to be something we're going to get into a little bit heavier here in a couple of minutes. Outside of doing enzymatic work or metabolic work, we can also see that they're going to be involved in muscle movement, oxygen transport, cellular structure, cellular communication, development of various types of tissue such as bone, and even within the immune system. They transcend all levels of the human body. They transcend all levels of the human body, hands down, without a doubt. And so we're going to see this concept of proteins over and over and over and over again throughout AMP1, throughout AMP2, into microbiology. It's going to follow you right on through. The other thing I do want to point out to you is the fact that enzymes are a major shape changers. Oops, let me try that again. They are amazing shape changers. That looks much better. In other words, they do alter their shape and their appearance for a whole host of various reasons that we're going to talk about. And that's actually why they can be as diverse as they are and do most of the things that they can do. All right. Here is where we needed to go as far as looking at this concept that shape is going to dictate function. What it looks like is going to tell us what it does. And so things like this guy right here, right? This here is often what we refer to as a binding protein. AKA enzyme. Right? This is your typical shape of an enzyme. And we're going to look at this structure a little bit more here coming up in a couple seconds. Because it is critical in our understanding of how enzymes work. The short story here is simply that we can see we've got this nice little open space right there. And it's within that open shape that other molecules can bind into. And once a molecule is bound into that opening, it changes the shape of the enzyme or the protein so that it can go ahead and do what it's going to do. We also have tunnel proteins or donut-shaped proteins. All right, tunnel-shaped or donut-shaped. We're starting to get into, and we've talked a little bit about in the lecture, about the fact that there are channel proteins that transcend the cell membrane. Well, this is the type of protein that you're dealing with. You're dealing with that basic shape, this tunnel or this donut appearance to the protein. We also have linear or fibrous protein shapes. We're going to talk about one of those key linear or fibrous proteins is actin and myosin that we will be getting into once we talk about muscle contraction. And then we have the good old globular proteins. This is this guy right here. Globular because, well, it's very blobbish in shape, if you would. Things like hemoglobin are globular proteins. There is a complexity that is tied into what these proteins look like and how they get to look like what you see in front of you. They don't start out looking like this. There is a journey by which all proteins go through in order to become either globular or binding or tunnel or fibrous in shape. And so in order for us to understand that, we have to kind of understand the journey that, or in order for us to understand the function and the shape, we have to understand the journey by which these proteins have undergone. And there are four basic stages in a protein's journey. We have the primary structure, we have the secondary structure, we have the tertiary structure, and we have the quaternary structure. These four phases of protein shape are symbolic of what they can and cannot do. Right? So, for starters, if we look at the primary structure of protein, we are seeing a linear shape or a linear form. It is simply a string of amino acids. That primary structure is a string of amino acids. It has taken the monomers, it has gone through polymerization, and it's now formed a linear protein. Not to be confused with the fibrous shape that we just got done looking at on the previous slide. Right? Primary structure happens right after protein synthesis occurs within the cell on the rough endoplasmic reticulum. This is something that Chapter 4 is going to get into very heavily. The other key thing with this is that it is not an active state. This is not an active state for this protein. It is simply a linear string of amino acids that have been bound together through the process of dehydration synthesis. That is all that we have here. Now, there is some form alterations that happen as we go from a primary to a secondary structure. We take that linear string of amino acids and we begin to fold the shape. Right? And as we begin to fold, we basically can create either a helical shape or a, hold on one second, which is very, I've lost my train of thought. still lost it. Helical, the spring-like shape, which is very slinky-like in appearance. Right? So it's actually kind of more or less looking like this as it moves down. I don't know how well this is going to work out, but we'll see. Right? That would be what we refer to as an alpha shape for the protein. Now, the beta tends to be a lot more pleated, and so it's going to look more like this in appearance. Right, so you can see the alpha here is a lot more spring-like, whereas the beta is more pleated, if you would, in appearance. Now, the key thing with this is that we have a protein structure that is still not active. And this is still a non-active form of the protein. It's not until we come down here to the tertiary structure where we even stand a remote chance of having protein synthesis become active. And the reason for that is because we have both globular proteins here and fibrous proteins. Fibrous proteins, again, are those very long, slender shapes that we saw on the previous slide. Things like actin and myosin, which are both protein filaments that function within muscle contraction. Both these globular and these fibrous proteins do have, and this is a key distinction, they can be active. They don't have to be, but they can be. And so this is the first stage where we really see the ability for a protein to be active. The last stage that we need to be concerned with here is the quaternary stage. And really, the quaternary stage is just an advanced structure of a protein. You take multiple globular proteins. Basically, you're taking multiple globular proteins and you're binding them together. And it's the bonding together of the multiple proteins that cause the protein to be activated. The key example of that is hemoglobin. And so you can see up top here, we have the primary structure of the protein. We see that spring-like shape there of the protein. That is our secondary structure of the protein. Again, primary and secondary are not active. And then we have the tertiary structure, which can be active. Again, this is going to be a globular shape that you're looking at there, but it could also be a fibrous or a linear shape. And last but not least, we have the quaternary shape, in this case here being hemoglobin. Remember I said that it's made up of multiple subunits or multiple globular proteins. Here is 1, here is 2, here is 3, here is 4, right there. And so there's your 4 subunits that have been bound together to create that quaternary advanced structure. Give me one second, folks. I am, all right, so as we come back here to enzymes, um, enzymes, as we mentioned, are special cases of proteins, and they do a lot of different things within the body. One of the key things that they do is they lower activation energy. They lower activation energy. Activation energy is the energy needed for a reaction to proceed or for a reaction to continue on and go. And so what the enzyme does is the enzyme lowers that energy, which conserves the overall energy within the body, and it also conserves the energy. Sorry about that, people. I was in the process of plugging in a computer and getting energy, electricity and power to my laptop. And so with the activation energy, think of it as, and I'll give you an example. It's right down here on the bottom of your screen. Here was the initial amount of energy, let's say, that is needed for a reaction to go. You can see that it is way above that line there. We incorporate an enzyme. The enzyme now reduces the amount of energy needed. You've saved that much energy. You only need now to supply that much. And so the enzyme is going to lower, it's going to lower the amount of energy that the body has to go ahead and put out for a reaction to occur. I use the analogy in class of an all or none concept with flushing a toilet. When you flush that toilet, there's a certain amount of pressure that you have to press down on the handle with before the toilet will flush. If you press enough, the toilet's not going to flush. If you press too hard, it's not going to flush any harder. Right? As soon as you get to that amount of energy that is needed for the toilet to flush, it's just going to flush, and it's going to flush normal. If you give it extra, it's not going to flush any harder for you. Same thing here with lowering the activation energy needed for a reaction to go. As long as you've met that minimal threshold for the reaction to go, it's going to go. Enzymes reduce the energy that is needed for that. One of the things it does is it speeds up the process. It speeds up the frequency of the reaction. Enzymes speed up the frequency of the reaction. This can be a very good thing, especially if you need a lot of energy and you need it in a hurry. And so you need to split up disaccharides or polysaccharides and break that down into monosaccharides so you can release the energy quicker. You don't want that to take a whole long time. The higher the activation energy is that's needed for a reaction to go, the longer of a delay in between each of the series of reactions. Enzymes shorten that break in between reactions and speeds everything up. Right? High activation energy is going to equate to slow reaction speeds. Lowering that activation energy is going to relate or to allow for quicker reaction speeds. Right? And so that is definitely key. Now, when we look at this bottom diagram right down here, here we see a typical enzyme right there. Right. The structure that we are breaking down in this case, which is here is sucrose, is referred to as the substrate. The substrate is what the enzyme breaks down. The substrate is what the enzyme is breaking down. And so we've got an enzyme and a substrate. And for all intent and purposes here, what ends up happening is the substrate fits down into the enzyme. It's a lock and key mechanism. Right? Only certain substrates can fit into certain enzymes. And so there is selection that happens here. We'll look at that on the next slide. And when this goes forward, what happens is the substrate locks down into the enzyme, and the enzyme changes shape and snugs itself around the enzyme, or I'm sorry, around the substrate. And once it snugs itself around that substrate, it can then break it apart, and it releases the product. And that's what we see here on this next slide here. The whole concept of a locking key mechanism is formally what we refer to as being specificity. Right. Specificity here, I usually tend to like to explain it as being I can't spell. Lock and key. Right. Only certain substrates are going to fit into certain enzymes. So when we talk about specificity, only certain substrates will fit into certain enzymes. And when we talk about fitting into a certain enzyme, we're talking about the enzyme's active site. Right? So for the enzyme right down here, the active site for this enzyme is right there. We can see right here is the substrate, right there, and that substrate is going to come down into the active site. When the enzyme and the substrate have bound together, remember I talked about there's that conformational change where it goes and it wraps itself around that substrate? There you're seeing it right there. Right? There you're seeing it right there. And so the the substrate is right there. So it's down inside of that enzyme. And from there, it can split it apart rather easily. Now, once it does that, it's going to release product. It's going to release product. Enzyme will split substrate and release products. Right? And those products are going to be simplifications of the more complex molecule that was released. Some enzymes will go ahead and just release the product, and then what it can do is it automatically resets itself and it awaits another substrate. Conformational change, snugs around it, breaks it apart, releases the product, accepts another substrate, snugs around it, breaks it apart, releases the product, accepts another substrate. Well, those products can either go right into being utilized by the cell, or in the case of most metabolic processes, it enters into what we refer to as a metabolic pathway. Right now, metabolic because it is usually an anabolic process where we've got the breaking down of a substance. Pathway because there's a series of steps that's used here. And so, as I tried to explain during lecture, we got a substrate that will go ahead and bind with enzyme A. Now, enzyme A is then going to release products. In this case, the product is alpha. Alpha being the product will then become the substrate for enzyme B. Alpha was the product of enzyme A, and it now becomes the substrate for enzyme B. Enzyme B is going to release product. In this case, it is beta, at which point in time beta will become the substrate for enzyme C. Enzyme C will take the substrate of beta, which is the product of enzyme B. Getting this now? The initial substrate goes into enzyme A and creates the products. Those products here would be alpha. Alpha now becomes the substrate for enzyme B, splits down alpha to create the products, which in this case are now beta. Beta, which are the products from enzyme B, becomes the substrate for enzyme C. Enzyme C is now going to break down beta and create products, lambda. And lambda is now going to become the substrate for enzyme D. And then the product of enzyme D is simply what we refer to as the final product. And so what we start with is definitely not what we end with. And it just takes us a series of steps to get from point A to point B. It's a chain reaction. And that's what we see here with this picture here, right, which is another way of looking at it. Here we have the initial substrate. Right, it's entering into enzyme 1. Enzyme 1 releases the product created from the substrate. And so substrate A becomes product A now becomes the substrate for enzyme 2. The product of enzyme 2 becomes the substrate for enzyme 3. The product of enzyme 3 becomes the substrate for enzyme 4. And the product of enzyme 4 is the final product. That's what the body is going to utilize. There is regulatory control over this, because what will happen is this metabolic pathway, these series of enzymes, as we know, will continue to go through and do this whole pathway. Because as soon as enzyme 1 is done, it doesn't just stop, it takes another product. It takes another substrate and will release more product. As soon as the substrate is out of the enzyme, it's accepting more substrate. Well, that can lead to a lot of dangerous excess of material that we don't need. And unfortunately, very often we do not store the products of metabolic pathways all too well. And so they just get broken down and wasted by the body. To try to cut down on the waste, what will very often happen is that end product that is produced, the end product that is produced, if end product reaches higher levels, what it will do is exactly what you see right here. It will actually go to enzyme 1 and shut down the enzyme. You can see right here, there's the active site for enzyme 1. Before being shut down, here's the active site for enzyme 1 now that it is being shut down. And so by restricting that active site, the substrate can't fit in there, and that shuts down the rest of the metabolic pathway. that metabolic pathway will go ahead and reinstitute itself once the levels of that end product drop below a certain level. Another way that we can stop an enzyme is by changing the environment, both temperature and or pH. And you can see here the process of altering the shape of a protein through temperature or pH is what we refer to as denaturing. And so what we've done here is we've denatured the protein. We've taken the protein, which is an active protein, it's in its globular form, and by altering the temperature or altering the pH, we're altering the shape of the protein. And we're actually sending it from, in this picture here, which is a tertiary structure, we're sending it back to a primary structure. And we know that primary structures are not active. As you change the temperature back into a homeostasis range, or as you increase or lower the pH back into a normal range, that primary structure of the protein will then become a secondary structure and eventually come back into being a tertiary structure and reactivate itself. A little bit on adenosine triphosphate. Again, the key things here that you need to make sure that you are aware of is that the energy within adenosine triphosphate is found in the second and the third phosphate groups. The majority of that energy is found in the third phosphate group. Right? Keep in mind that adenosine triphosphate is the energy of the body. That is what your cells, that is what all of your metabolic reactions that require energy are using. And so adenosine triphosphate is critical for the proper functioning of the body. And the other thing is, the body only has the ability to really store about 30 seconds. I misspelled storage. I apologize. But it only has about 30 seconds worth of ATP that it has on its reserve. And so being able to constantly create ATP is critical. Where ATP releases energy is within those second and those third phosphate bonds. And so if we look here, this is a typical structure of ATP. We see we have a sugar, and that sugar is ribose. You can see here that we have a nitrogen base, which is adenosine. And we have the star of the show, which are those three phosphate groups right there. This phosphate right there, that's the third phosphate, has the highest energy. This is your second phosphate. And it's got lower energy. And then your third phosphate, which actually becomes the first one, this is your first phosphate right here, that's got the lowest amount of energy. So much that it doesn't even break off and release any energy. The energy that we are using to run cellular metabolism is happening right there, between the third and the second phosphate. And so you can see right here in this diagram here, we are again focused on that phosphate and that phosphate. At this point, we are dealing with ATP, adenosine triphosphate, but at this point we are dealing with ADP, adenosine diphosphate. phosphate because we just have 2 phosphates left. Right now, if we do in fact release that second phosphate group, then we go down to AMP, which is adenosine monophosphate. So when we talk about energy, when we talk about proteins, when we talk about enzymes, this is what we're referring to. What I would like to do is spend just about maybe 5 minutes or so looking at lipids. And then we can call Chapter 2 done, signed, sealed, and delivered. And so, switching gears, this is the final macromolecule. Keep in mind that we've really talked about 4 macromolecules. We started out talking about carbohydrates, then moved to nucleic acids, spent a great deal of time talking about proteins, and now the fourth, that being a lipid. A couple key things. All lipids are made up of carbon, hydrogen, and oxygen. Just like carbohydrates, the key here is less oxygen. Right? That's really the key here. The amount of oxygen found within lipids is much less. You have a much higher presence of carbon and hydrogen. Right? These guys are always nonpolar. In other words, they are not going to be mixing all too well with water, which is a polar molecule. And if anything is going to dissolve in water, it needs to be able to have positive and negative ends. You're not going to find that with the lipids. So therefore, because they are nonpolar, we refer to them as being hydrophobic. And the great majority of our fats really come in 2 key structures. We have the hydrocarbon chain, right? Hydrocarbon chain, it's a chain because we've got a series of carbons that are linked together. Hydrocarbon because, look, it is surrounded by hydrogen. It is surrounded by hydrogen. Then we also have second part of a lipid, which is the carboxyl group. Right, now that is a very familiar looking structure, that carboxyl group. We know carboxyl groups are defined as having a double bound oxygen as well as a hydroxyl group or an O negative. Almost all the time when you see a fatty acid, as what you're looking at here in front of this, on the screen, it is almost always going to be C double bound to oxygen, single bound to a hydroxyl group. It's only during dehydration synthesis will you see an O negative. We have mainly 2 types of fatty acids that we're dealing with. We have saturated and unsaturated. With a saturated fatty acid, you will notice that all carbons are surrounded by hydrogens. All carbons are surrounded by hydrogens. That is the definition of a saturated fatty acid. It is saturated with hydrogen. An unsaturated fatty acid, on the other hand, is not completely surrounded by hydrogen. In fact, if you look right here, you will see the culprit. There is at least 1 double-bound carbon in an unsaturated fatty acid. Because each carbon has a maximum of 4 bonding sites, right? Remember, each carbon has a maximum of 4 bonding sites. 1, 2, 3, 4. Right. Every single carbon has a maximum number of bonding sites of 4. Well, this carbon right here, there's 1, there's 2, There's 3. There's 4. There's no more room for carbon to bind to any more hydrogens. And so that is why we have that missing appearance, or that unsaturated appearance for unsaturated fatty acids. Real quick, very often we can have triglycerides, diglycerides, which we know from looking at health studies, triglycerides are very unhealthy for us. Right? And basically, when we define a triglyceride, what it is 3 fatty acids. Basically, what we're dealing with here is 3 fatty acids, which we know fatty acids are going to have the hydrocarbon tail with a carboxyl group that's going to be attached to the top. And those 3 fatty acids, if it's a triglyceride, is going to be bound to a glycerol group. Now, here's the structure of a glycerol group. Here's your glycerol right here. Right. Glycerol groups are defined as characteristically having 3 carbon molecules, which you can see right here. There's 1. There's 2. There's 3. And those carbon molecules on the top are going to be surrounded by hydrogens right there. But on the bottom, we do not have hydrogens. Instead, what we have are 3 hydroxide groups. We have 3 OHs that are found right down there. When those hydroxides come in contact with the hydroxyl group associated with the fatty acid, dehydration synthesis occurs. Dehydration synthesis occurs. And if I was to be able to move that, and so if we look right in this area, right here, you can see that you've got a hydroxyl group right there for which the hydrogen is going to be donated, and we've got a hydroxyl group that's associated with the fatty acid right there for which the hydroxyl group there is going to be donated. Hydrogen from the glycerol, hydroxide or hydroxyl group from the fatty acid, H2O, water is released. And we create these nice, neat little bonds that are formulated right there. And that is what a glyceride is. That is what a glyceride is. Whether if it's a triglyceride, it's got 3 fatty acids. If it's a diglyceride, it's got 2. If it's a monoglyceride, it's got 1 fatty acid that is attached. Steroids, all right. The key thing with this is what alters the type of lipid what alters the type of steroid that you are dealing with is the side chain. Once again, all steroids are going to be composed of 4 rings. Right? It's the functional group. It's the functional group that is going to differentiate one steroid from another. And again, you've got a polar region which is able to interact with water, and you've got a nonpolar region which is not able to interact with water. And the last thing I will say is that another type of lipid that we have is what we call a phospholipid. We're going to talk about this much more in Chapter 3, which will be the next WIMBA session. And with a phospholipid, we have a diglyceride. Right? So it's a glycerol with two fatty acids that have attached to it. But then sitting on top of it, we have a hydrophilic phosphate group right there. And so the head region, which is characterized by the phosphate and typically a choline group, which just enhances cell communication, that there tends to be polar or hydrophilic, whereas the diglyceride, the glycerol group with two fatty acids, tends to be hydrophobic. And therefore it makes it non-polar. So that was kind of a quick review of, or introduction into what lipids are and the type of diversity that we can have with them. There is a lot of information that we covered within these three WIMBAs. I would encourage you to go back and really look at and listen to these three WIMBAs on biochemistry, 1 of 3, 2 of 3, and 3 of 3. And if you have any questions, please make sure you jot them down. Shoot me an email. Let me know what's going on. Don't let what you think you understand go by. If you have any questions, ask. So that way we can correct things as we go along before you get to the exam. And on that note, happy listening, and I'll see you either in lecture or lab.